to Suzanne.
Sure. Thanks, Lindsay. Sorry we're a few minutes late. Always traffic on 76. My name is Allison Minarovic. I'm the collection manager of the diatom herbarium at the Academy of Natural Sciences. I also used to be the project phycologist for the DRWI. I spent my first 2.5 years working on that. This is Catherine Christopher.
Yeah, you guys, if you went to Watershed 101, I may have already met you. My title on there now is Science Communication Specialist, but first and foremost, I'm sort of the first point of contact for all the DRWI cluster people, especially regarding monitoring and other kinds of support. So I wear many hats, but I do a lot of field work and a lot of monitoring support. So.
I'm Amanda Chan. I work with the DRWI project. I help mainly with the algae sampling for the past couple of years, and used to work with Frankie when he was with us.
Yeah.
So if any other questions, I feel like we're set for it.
Yeah. And thank you all for being here today. This is a really great turnout for volunteers. If you have any questions during the presentation, please feel free to interrupt us. No need to save the questions for the end. This is for you guys, and we want to make sure that you get the information that you need. So we're presenting today on the visual algae assessment. This is for your monitoring, and ideally you'll be doing this either during your chemistry assessments, if and when you do that monthly, and or during the habitat assessments. Ideally, you'll be doing this monthly. If you're not able to get out that often, a good time to monitor your stream is early to midsummer. So you should have the algae assessment in front of you. At the end of the presentation, we're going to go through each part and just kind of break it down. But first, we'd like to give you some background information on algae.
And actually, if you have any questions in the meantime that come up, please interrupt us. It'll just be easier to answer them on the fly.
Yeah. So first, the reason why we're monitoring algae in the first place, algae are really great indicators of water quality. Unlike fish or macroinvertebrates or monitoring chemistry at one point in time, algae kind of represent a short period of time. They reproduce a lot faster. They can also live in any aquatic environment where you maybe wouldn't see fish or macroinvertebrates. They exist year-round, although there is some succession, and you'll find certain algae at some parts of the year and others during other parts. They respond a lot to nutrients, oxygen, because they get their energy from the sun, and they need oxygen in order to reproduce. They also respond to changes in pH. If you have really acidic conditions, you'll find certain types of algae. If you have really high pH, you'll have a totally different community of algae. Some of the other things that they respond to are other types of pollution, whether that be metals, stormwater runoff, salt, things like that. And there are certain species that are really sensitive to these changes, and they're considered to be indicator species. So some of these you need to use a microscope. Others you can see blooms of different kinds of algae, and we'll go through that later in the presentation. Each species has very specific requirement for their habitat, much like humans do, and then there are others that can exist in just about any condition. So I'm sure that you guys have some concerns about algae, what a lot of algae means, if that's harmful to your stream, and whether it's good or bad. Some of the things we hear most often are complaints, or they impact recreation, or they just look and maybe smell bad in your stream. If there's a lot of growth, if you think about a lot of nutrients going into your stream, that's kind of like fertilizer for algae, and that allows them to grow really fast. As they decompose, they can also release smell. It might affect the taste of water. Hopefully, if you're drinking tap water, it will be properly treated, and a lot of that odor and taste problems will be removed. So some of the things that we're going to ask you to look for as you do these assessments are nuisance algae. So you'll see on your paper towards the bottom there's something called rocksnut. This is a type of diatom that is considered to be invasive, and we'll show you some pictures of what that looks like later on in the presentation. But it's
Not a
unlikely.
technical term.
Yeah, it's there's a certain species name, but the common name is rocksnut, and there's a reason why it's called that. It's unlikely that you'll find these blooms, but we want you to be aware of any red flags. Other things that we are concerned about are blue-green algae blooms. If you've ever seen satellite photos of Lake Erie or like really thick paint-like lakes mostly, those are blooms of cyanobacteria or blue-green algae, and they have the ability to produce toxins. But in rivers and streams, we rarely find that because they like standing water, because the water's flowing. If you see any blooms of blue-green algae, they're probably not harmful. And because algae are the base of the food web, they're the food for macroinvertebrates and fish and higher trophic levels. So if you have major changes in algae, you have really large blooms of algae that can block out sunlight, that has major implications for higher trophic levels, things that would eat algae, of course. So things to consider before you go out and sample. It's important to think about why you want to sample and what your major concerns are. You want to think about what your reason is for monitoring. Do you want to look for changes in the stream that you are assessing? Are you concerned about certain things like the smell or major blooms, unsightly conditions, or seeing fewer fish than you would normally see, or just anything that looks different than what you're used to? You also want to think about what the best method is for your needs. This visual assessment is a really great way to track changes in a rapid way so that you're not spending a whole lot of time going into the stream and looking at finer details. Timing is also an important issue. As I mentioned before, there's seasonal succession of algae. So you'll see some kinds of algae earlier in the growing season, so early spring, through the end of fall, you'll see different colors and different blooms. To assess yearly trends, it's important that you sample at the same times each year. So if you're sampling monthly, you would want to sample roughly the same time periods, or at least like allow for the same period of time to go by. All right, so this is what the seasonal succession of algae looks like. In the early spring, you have like thin, like crusty algae that grow on rocks or substrate like sand or even decomposing wood or aquatic plants. It's not a whole lot, and that's because we're just getting over winter, and a lot of algae don't prefer really cold conditions. As you move forward in the season, you'll have some what are called prostrate algae. They grow a little bit more heavily on substrate, and you can see them more as just films. A lot of this algae looks this way, and there's not a whole lot of it because of macroinvertebrates feeding on the algae. As the seasons progress and you have early summer, especially if you don't have a lot of major storm events, this allows time for different algae to grow stalks, kind of like how plants do. They have short stalks and start to form communities or filaments of algae. And then over time, you get longer filaments of mostly non-diatom algae. So anything that looks green or blue-green, yellow-green, things like that. And you'll find longer strands of algae. Over time, these will start to die off, and filamentous algae will start to grow on other plants or moss or, other things that are growing on the banks of the stream, like roots. And then closer to the end of the summer and early fall, you'll see like gelatinous globs of algae. And we'll show some pictures of what globular algae look like. So it's important that you understand these patterns because these are normal conditions. Sometimes you'll find really major blooms, and that might be indicative of major nutrients flowing into the stream, or maybe some kind of other concerns that seem unusual. There are some physical and chemical forces, other pollutants that can affect algae growth, but some of the major things to consider are major storm events that can scour away all of the algae that have been growing on rocks or sand or other substrates. If there's a lot of sediment flowing through as well from storms, that can also scour away algae. It can also lead to really turbid conditions, so really cloudy streams, and that blocks out sunlight, so it makes it harder for algae to grow. Again, with storms, you have really high flow rates, and we recommend that you wait at least 10 days, if possible, to study or to do your assessments, so that it goes back to sort of baseline conditions. Yes?
So, is some algae in the stream good, desirable, or like some of it, like you want some of it, but some of it, but you don't want too much of it? Like what's algae good, algae bad?
Yeah, that's a great question, and I should have addressed that at the very beginning of the presentation. In general, algae are not bad. Algae are food, and they're the base of the food web. If you have too much algae, like you have major blooms, and it cuts off sunlight and depletes the oxygen in the stream, that can be bad. The algae themselves are not bad, but they respond to things that are flowing into the stream that are bad. So they are indicator species, basically. And that's a sign that there are other things going on, point or non-point sources of pollution. But in general, algae are not bad. And not all blooms are indicative of bad conditions. Sometimes that's natural, and it really varies between streams. If you monitor a really shady stream that has fast-flowing water, mostly rocks, and it might be really shallow, and you see a lot of fish, you may not see a lot of algae growth, and that might be your normal baseline condition. If you're monitoring a stream that is slow-moving, has steep or vertical banks, and it's like a sandy bottom, maybe it's near a farm, you would see a lot of primary production or a lot of algae growing. You can think about it like a lot of fertilizer maybe from the farm going into the stream. And that might be your baseline condition for what normal amount of algae look like. And this all has to do with temperature, light, how fast the water's flowing, and how many other organisms are in the stream that feed on the algae, because grazing can have a lot of effect on how much algae grow in your stream. So here's an example of a visual assessment pre and post scouring or major storm event. So the three pictures on the top are normal conditions, and it may be a little bit hard to see, but there are sort of thick films of brown and green algae growing on all of these rocks here. And then after a scouring event, you can kind of see that some of these films or mats of brown algae, have been scoured away. So this is an example of a major storm event, whereas this would be sort of heavier growth. So like on the higher end of medium, low end of high, and then this would be much lower. So maybe like 1 to 4 if you're using the visual assessment and you're thinking about how much algae is normally in my stream versus after a flushing event. So that's why we recommend waiting 10 days in between storms, if possible. I know that some years it's especially rainy and you can't always predict the weather.
So in this picture, which of them you consider good condition, which consider bad condition?
Well, it's not so much a matter of good or bad. If this is what you're used to seeing, and it's been 10 days or 10 days to 2 weeks, or if you've had just really sunny, nice, warm weather for a while, this might be what you're used to seeing. But after a storm, sediment and rocks may move, and it flushes away a lot of the algae that are normally there. So it's just different.
So you want to say that algae will be brown and the green one is when there is no algae?
Well, it's more that this is just showing before and after.
Yes,
These-
but I don't understand what the green color comes from.
They're different types of algae, and we'll go through examples of each of these, but the brown algae, looking for a pointer, there is one. Here it is. So this area right here, and over here, down here, these are all types of brown algae, or golden brown algae, and those are mostly diatoms, especially in freshwater. There are other types of brown algae, like kelps or marine species, that you're not going to find in freshwater streams. The green algae over here, these are just different types of algae. They can either form thin films or thick mats or long filaments. And then down here, you mostly have just a little bit of brown algae, maybe a teeny tiny bit of green, but if you're doing a visual assessment from a stream bank, you might not see this small amount of green algae.
So you're saying that on the bottom picture that algae was washed away. That's what you say.
Yeah, so if you think about it on your scale of 1 to 9, if this is, if you are routinely sampling your stream and this is what you're used to seeing, this would be an example of a 5, maybe, just as like a middle of the road baseline. And then this, if this is the least amount of algae you've ever seen in your stream, you could rate that as a 1 or a 2.
But it's important to note how many days it's been since a large rainstorm. So then you can tell yourself, well, there's not as much algae as I'm used to seeing, but we did just have a huge storm yesterday, so I should wait another couple of days before I reevaluate the health of the stream using the algae. Whereas this is just saying, nothing's wrong, it's just, it all got washed away. So it's a good before and after picture. It's a good photo to show different levels of thickness or thinness, but it's also a reminder to say, like, a storm can have this impact, and so that's why the protocol is sort of to wait after a storm event. It wouldn't be if you went out right after it rained and did your assessment, it wouldn't really be a true assessment of the stream. And that goes for all of our sampling as well, and especially in terms of algae and macroinvertebrates. Things move after a storm event, and chemistry too. Base flow, we kind of just need to wait for things to get back to normal before we can use our one sampling event to categorize the stream.
Okay, so overall your recommendation to do the monitoring like week, 10 days after major.
Yeah, if possible. And that's how we have been sampling during the DRWI. If you are sampling on the same day each month, you might not have any control over the weather, and so you might, on your weightable streams assessment, that first page, you may write 1 or 2 days post-storm event, and that's a good way to track how much growth occurs over a period of time. I saw a question back.
Yeah, is that greenish hue on the bottom indicating that it's phytoplankton?
This is actually just photos taken from the top of the stream. It's not the best quality picture because it's not totally clear water. It's mostly just turbidity, dissolved organic matter. But in rivers and streams, you'll have very little plankton unless you have really slow-moving water. Most of the algae that you see are called benthic algae, where they stick to surfaces. And with flowing water, you need to have special parts to your individual cells that allow you to stick to substrate. So you'll mostly find plankton in lakes or pools within a stream. Does that answer your question?
Yeah, I was just wondering why it was green.
Yeah.
They're probably from the same site and on different days, and so it could even just be like maybe it was sunnier that one day and it just made the water look different.
Yeah.
Yeah.
Or or fresh after a storm, and this
Yeah.
is just still sediment that's waiting to settle.
And I think they're taken using this sort of viewer, and I don't know the name of it, but it's kind of like a cone-shaped thing with a clear bottom that you push into the water to see clearly the substrate. So I don't think those are taken with like an underwater camera. I think they're taken using that device.
Yeah, if you have a clear bottomed large
Like a bowl,
petri dish,
yeah.
a bowl, a storage bin, and you want to take pictures of big blooms of algae or look at the thickness, you can just push that into the stream, get it just below the surface, and take a photo. And if you don't want to take like a stream-wide photo, it's a good way to get up close photos of getting your phone or your camera wet.
Excuse me, could you repeat that?
Oh, sure.
What you can use to do that?
Anything with a clear bottom, whether that be a clear storage container, maybe a piece of glass or like a plexiglass thing that you can just stick in, a big petri dish, or even just a clear bowl would work, something glass. Yeah, doesn't have to, there's no technical equipment really. You know, when I go out and collect algae, sometimes I don't have anything to collect with, so I'll just look for a bottle that has gone into the recycling bin, rinse it out, and collect some algae from there. So our tools are very primitive. Okay, so this is an example of nuisance algae. So really, really thick blooms of algae that may be unusual for what you're used to seeing. So if this is your baseline condition in your stream, this would be a 5 on your scale. This would be something like a 9. This has a lot of filamentous green algae. There's some brown algae that's probably underneath. The rocks, I mean, you can't even make out the outline of the rocks. You see like thick brown growth with a lot of like green filaments on top. So that can affect the flow of the stream if you have a lot of really thick growth of algae. And if algae get washed downstream, it can form even larger mats. This also makes it harder for fish and macroinvertebrates to move around in a stream. And this last bullet point mentions a genus of blue-green algae called Formidium. And there's a little bit of that in here. It's hard to tell because of the color, but unless you see really obviously blue-green algae, like you'll see this bright turquoise color, you could consider this brown algae. Formidium is not a bad genus of blue-green algae. It's not toxic, and it grows there normally. Blue-green algae are some of the first algae to colonize a stream, and they've been around way longer than other types of algae. They're very primitive because they're bacteria, but they're really important. They're really important for forming that initial biofilm upon which other algae can grow. So some of the reasons why you'll have high versus low biomass, we touched on this earlier in the presentation, but algae in general respond to nutrients, light, and temperature. They're just like plants do. They're aquatic plants, they get their energy from the sun, and they want fertilizer, they want light, and they want nice warm temperatures. They don't really like to be cold, just like we don't, or they don't like to be too hot. Some of the things that can cause a loss of biomass are scouring, big storm events. If the substrate's moving from really fast-flowing water, sometimes caused by storms, that can dislodge any algae that are growing on different substrates. You also have grazing pressure from other organisms that eat algae, so that can reduce the biomass. So let's talk a little bit about the visual habitat assessments. So this is the first page that you guys fill out when you go and monitor a stream, and this is really important because it connects to the visual algae assessment. I'm not sure how often you fill out this habitat assessment, or if you fill it out at the same time as your algae assessment, but there are some parts that would...
We've never seen
If you happen to have access to additional copies, you wouldn't need to fill out the entire assessment because there are a lot of things about your habitat that don't change. But if you're doing your visual algae assessment, it's important to note the weather, whether it's clear, cloudy, overcast, and how many days since the last rainfall event, because that can give you some information on why there's either low or high amounts of algae. You wouldn't need to fill out this part because you are already mentioning it on your habitat assessment, or your algae assessment. And then these water conditions down here will probably not change. If you notice any changes, mark them, but your habitat assessment should be relatively stable each year. If you only have one of these sheets per year, please just mark on the back today's weather and days since last rainfall. And any other conditions that seem out of the ordinary to you. So here's your rapid algae assessment report. This is what you should have in front of you. If you don't, please raise your hand and you can get an extra copy. Awesome. So this is not meant to take more than 10 to 15 minutes. It's supposed to be rapid, and it's supposed to be you looking from the stream bank. You shouldn't have to get in the water unless you want to. That's fine. And if you want to take pictures from the stream bank using a glass bowl or a storage bin. So we're going to go through each of these sections one by one. You're first going to fill out your name or ID of the stream that you're monitoring, the date that you're sampling, and also the time, because morning, afternoon, or evening, you want to make sure that you're trying to sample at least the same time each time you sample. You want to mention who you are, and after you submit your algae assessment report, Lindsay or another person will complete your review. And this review will be based on the photos that you take, which we'll talk about at the end of this assessment. So this next part, we're trying to break it down by color. We don't expect you to know exactly what genus or species you're looking at, because again, you're looking at algae from the stream bank. So we're breaking it down by color. Brown algae are mostly diatoms, and these are the most abundant algae that you'll see. And if you see any of the following types of algae, if you see any of these colors, just check the box. You want to consider a 5 to be your baseline condition. So whatever you're used to seeing. If you have lower than expected algae, you will circle anywhere from 1 to 3. If you see slightly lower than what you're used to seeing, you would mark a 4, slightly higher is a 6, and then 7 through 9 is a heavy amount of algae of each color. And then you want to note what it looks like. And you can circle as many different growth forms as apply. So if you see filaments, and we'll go into the stream and also look at some pictures. Sorry.
So, I know you said algae's in the stream all year, but do you want volunteers to do the assessment in the spring, like in a spring to summer period? Because doesn't the baseline amount of algae change like throughout the year?
It does, and that's up to you guys how often you want to sample. If you have access to the stream and you want to look weekly, that's fine, because you may not necessarily want to fill out an assessment, but it would give you an idea of changes over a shorter period of time. But it's really up to the group and what protocols you would like to set. Yes?
Are you saying that what we should consider the baseline is our, say, June measurement? My site is full of, it's covered with trees. I often see the most dense algae in February if you have a warm-up and there's no leaves on the trees and there's boatloads of light.
Yeah.
And sometimes it actually is less, for example, during June because the canopy's covered over and there's less light. But should I be using June as my baseline, for example, then? Because it does change the year.
I would ask Lindsay about that. How often do you you do the habitat assessments once per
a month.
year?
Once a month.
Once a month. And you sample chemistry
Not
once a,
anymore.
sorry?
Not anymore.
Not anymore, okay. I would fill out after one calendar year of habitat assessments, record your algae assessments each time, and use your baseline condition for the middle of what we would call the growing season, so the middle of the summer. Because in some cases, in the winter months, like late winter to early spring, it may be unusual to have no leaves on the trees, lots of sunlight, but during the middle of the summer is when we normally expect the most algae growth. So for you, that would be a lower amount than, that would in the late winter, early spring, that would be a high amount of algae compared to your summer baseline.
So all this training, what I'm hearing, you recommend to start doing this monitoring in June.
Yeah, so I believe that's the next time that you're sampling? Or you're doing your assessment? You're doing a spring?
Well, what's the Hickens, the 10th to the 12th? I mean, the 10th through 20th.
They do, each month, there's a window of time. Later in the month, you said like the 10th to 20th? So if each month, they'll go out sometime in that window.
Okay, so you're about to fill out your April assessment right now.
Correct.
Okay. In that case, it would probably make the most sense, unless you're really familiar with your stream and you know roughly how thick your algae growth is in the summer, maybe start with April as a baseline for the time being. And each month, if you're sampling year-round or just during your sampling period, you can recalibrate and see how much greater your algae growth is in the summer. And then the following year, you can use that as your baseline. If that makes sense.
Would make this normal baseline keep changing? I mean, everybody has a different interpretation. Depends on when, where, all that you just explained. Would it make more sense just the picture you just showed? We find an area that's more representative and see the coverage on the rock. Like in the picture you showed a very clear, so it's almost 100% of coverage. Some just gave up 2 or 3 buckets. Would it be more comparable over time?
It's actually site-specific. So, for example, if you have a really shaded stream in the summer, you would expect your normal baseline condition to have a low amount of algae. So you want to calibrate it to the stream that you're used to seeing and that where you are sampling. So it'll be a little bit different for everybody because every stream is a little bit different.
And you guys know your streams better than we do, obviously. You're the ones out there seeing it all the time. So it's kind of, we're looking to you to tell us what's normal and what's not. And then, as you go on, be the eyes and ears and say, okay, now this is different than how it usually is. Because it is very site-specific. So, I mean, we know the sites that we go to, but one small creek could be very different from the one down the road.
And the other technical question, you gave us this form. Is there a way to fill it online? You
Yes.
provide that information?
Well, I mean, it will be on the Street website.
Okay.
TCF.
Okay.
And we can share the presentation also.
Yeah, of course.
Does the same algae turn color? I think around January, when I observed my stream, it was bright green, which it's never bright green. And then the next month I went, it was almost black, that
Okay.
brown-black color. Is
Yeah.
that the same algae that just turned that color, that's dying off because it's wintertime? I mean, I think it might have been, I do measure phosphates, and it might have been overlapping when there was a high phosphate level. But it was bright green, which is very unusual, and then it was, the brown-black. Is that the same algae?
So when you were, and if you remember back in January, were you seeing a lot of green algae, like the bright green algae? Was it more like filaments or kind of just like a coating of film of green?
Probably more coating.
And did that same coating turn brown and black, or did it look like different growth forms?
I have have no clue.
Okay.
All I know it was brown black then.
In that case, it's probably better.
I'm near a processing, a sewage treatment plant
Oh,
downstream from
okay.
the sewage treatment plant.
Ooh.
And that probably alters the temperature somewhat and the
Yeah.
nutrients going in.
Definitely. Usually when algae die, they will no longer be able to stick to whatever substrate they're growing on, and so they'll be washed downstream. So I think that the best way to do that is to go just based on visually what color you're seeing, because it might be turnover and a new type of algae. It's hard to say exactly what that is unless you're looking at it under a microscope, but I would think that if it is dead, it's probably
It's probably gone. And
Yeah.
that's the normal algae is what was there.
Yeah. So you want to, in addition to checking off what colors you're seeing and marking how thick of growth you're seeing of each type, you want to also circle as many different growth forms as apply to whatever stream you're looking at. So for brown algae for example, you may see mats of golden brown, brown-colored algae growing on the surface. And these would be just like thick chunks on top of the water. They're probably moving along with the stream. If you're seeing filaments, and if you have the ability to reach down into the stream, these will be really light, flowing filaments that kind of look like thin hair, but if you pick them up out of the stream, they'll break apart in your hands. There are also cases where you have filamentous green algae that are covered in brown algae, and we'll show some pictures of that a little bit later. Yes?
So are the filaments rooted, or are they anchored in some way in the stream?
Yeah, they'll be attached to rocks or other types of substrate. They won't be flowing down the stream. Under the surface, if you're seeing any algae floating on the surface, you want to either circle whether it's thick or thin. If it's a really thick mat, or if it's just like a thin layer, different than a surface film. A film would just be a really, really light coating. It may look like a layer, think like pollen, just like that thin, forming a film. So under the water, you may have submerged mats, and these were some pictures that we saw a little bit earlier with like really thick brown growths on rocks or other substrate. You may see tufts of algae, something that looks like it's growing up, and this is different than the flowing filaments. You may see big globs of algae that look different than maybe like softer looking tufts. And this is subjective. This is how you would perceive what's growing in your stream. You may see something totally different than any of these growth forms that we may have missed. There's all types of ways that algae can grow. So if you're seeing something that's unique or doesn't fit any of these categories, just describe what you're seeing at the bottom. And we have the same growth forms for each of these different types of algae. If you see something other than brown or golden brown algae, or green algae, or blue-green turquoise algae, please mark it. And down here, in addition to mentioning the growth form, please just describe what colors you're seeing. There are other types of algae, like red algae, for example, that are unusual to grow in freshwater. Most red algae are marine species, but if you're seeing any red colors, we would want to know about that, especially if that's not what you're used to seeing. So, some of the things to look out for, and we wouldn't expect you to see many of these following things, but these would be sort of red flags, things that are really unusual for your stream. And this would be important information for us to know and for you guys to know as a group if there are any streams that may need closer monitoring. If you rank any of the following categories of different types of algae as a 9, that's really, really high, unusual amounts of growth. That may indicate a lot of nutrient or other types of pollutants in your stream. So you would want to know about that. Do you see any rock snot? And we'll show you some pictures of that. Rock snot is a type of diatom that if you pick it up out of the water, it feels like wet wool. And under the water, it looks like dirty, wet toilet paper, kind of. There's a lot of mucus in this type of algae, and it's golden brown in color. And if you see any large films or blooms of blue-green algae, we would want to know about that as well. It's really rare that you would see that in a flowing stream. In fact, in all of the years that we've sampled for the DRWI, we've never seen blooms of blue-green algae. Maybe some films growing on rocks, but in very small patches, or just a few globs, but it's really rare that you would see that.
Have you seen rock snot?
We have not. We've seen blooms of diatoms that look like tufts or globular forms that we weren't sure if they could be rock snot, so we ended up taking them back to our lab and taking a look at them, and they ended up being a different type of algae.
I believe that, I've heard that it is in the main stem of the Delaware on some parts, but not in any of the small tributaries yet where we sample, but we still take precautions if we know that we're bouncing from stream to stream on any given day, or especially watershed to watershed, if we do multiple sites per day. There are things you can do to prevent the spread of not only Didymo, but other types of things, is we just clean our waders off in between sites, but that's more of just going above and beyond, because again, we haven't seen it.
Didymo or rock snot prefer really cold, fast-moving water, and if you're monitoring during the spring, summer, and fall months, you probably won't see this. You probably won't see this in any of your streams in general, especially around the Philly area, because the water tends to be a little bit warmer. But if you suspect that you see it, please let us know. This is important information for us to know.
There's a photo of where I thought that. Yeah, we have a bunch of pictures coming up.
Yeah. And we'll explain more about why this is a red flag in greater detail. And finally, we ask that you take up to 4 photos of any big clumps that you see. If you see higher growth or less than your normal baseline condition, and any just general photos, these can be up close in stream, or it can be just photos of the stream that you're monitoring in general. Both types of photos would be good information. That way you can track different changes over time, and you have something to compare to your visual assessment.
Pictures that you mentioned, this is kind of addition. We usually supposed to take 3 pictures of the stream. You are saying that these pictures only we see something unusual.
Those are the most important things to take pictures of. If you don't see anything unusual, still take up 3 to 4 photos of your stream, just as...
That's the generic pictures that we always take.
Yeah.
Okay.
But especially if you see anything unusual, you would want to document that. If you see a lot of unusual growth forms in addition to your normal 3 photos, you can take additional photos and just record that on the back. But I think 3 to 4 is a good number of photos. Yes?
I think that the question I have is that I understand there's 3 photos that we normally take upstream, downstream, and then across the stream. But is there an additional photo that we want to take on a regular basis that's just of the algae condition?
Yes. For the algae assessment, you would want, if you're taking upstream, downstream, try and capture in-stream as well.
With this plate thing, with this plate technique.
It doesn't necessarily have to be that. If you're interested in getting a closer look, that would be great. Otherwise, we just want to see, in general, the overall coverage.
And we have photos here that we took of our streams, which were the ones that are you take upstream and downstream, but you can also clearly see what's growing in the water too. It just depends, if there's a lot of glare and the picture doesn't come out well, it's not going to really show as much of the algae. So
Right. So these photos would be focusing more on that algae. Yes?
maybe you're gonna get to this, but how and where are we gonna submit this sheet and the photos to them?
To your normal watershed, volunteer coordinator.
Then is there an online form where we fill
Well,
this out?
yeah,
Oh, we'll
we'll-
get to that?
Yeah, we'll get to that.
Thank you.
Yes, I just wanted to make sure I'm understanding, filling out the form in terms of a baseline. We just started monitoring my stream this past November, so I haven't seen a summer version of it yet. So if the middle of summer is supposed to be baseline, how do I fill out the form until then? What do I call baseline? Just what I was seeing the month before? Or-
Yes, I think that a good way to start, because you're in the middle of your April sampling, is to start with April as your baseline. And for everybody, if you're unfamiliar with what baseline conditions look like in the summer, start with April as the baseline, and after one full year, you can recalibrate.
Okay.
And then recalibrate the summer, like June or July, as your baseline conditions. And that would be something that you would all agree on as a group, whether that's June or July.
And I know I realized we didn't really put a spot on here for miscellaneous notes, but maybe it just would be helpful to jot down, like, this is the first algae assessment I'm doing, or, just a little comment about how you're feeling about it.
Yeah. And on the back of this form, you're welcome to write as many observations as you feel necessary. Of course, we're all scientists,
Yeah.
and taking notes and observations is a big part of why we're doing assessments, in addition to the things that you're marking here. All right, so any other questions about the assessment? At the end of, I'm sorry.
Maybe you're gonna show this in the pictures, but most of my algae is gonna be like what you saw prior, where it's just like covered in rock.
Uh-huh.
And I'm not sure whether that applies to the thin or the thick.
Again, that's subjective, so it's really up to what you're used to seeing.
Okay.
And every site's going to be a little bit different, so it's more of your own calibration for your specific site. Okay. So at the end of this presentation, we're going to do a practice. So we're going to have a baseline condition and a second photo where we're all going to fill out one copy of, do we have that?
We have a photo of a site, and we're gonna say, how would you guys fill this out? And then we're gonna see how that coincides with what we would do.
Okay.
Yeah. Just go over that.
Yeah, we'll just do a practice run and see how people would fill it out.
And, when we have time to get down to the water, we can do the same thing.
Yeah. So here's some of the algae that you'll probably see. Especially in the springtime, there are large blooms of green algae, and these kind of slimy filaments of green algae, like over here and over here. They look really shiny. If you pick them up in your hands, they feel slimy. And then there are also sort of like thicker, like scratchier forms of green algae. And these are all filamentous. You can see that they kind of look like really thin threads here. And this is an example of what you would record as green filamentous algae. And as just a quick visual, what you see in the stream in general, you would want to mark whether it's more or less than approximately ½ meter in length. It doesn't have to be an exact measurement. We are absolutely not expecting that, but just as an idea of how much filamentous algae growth you see. So here's an example of green algae, and this applies to brown algae, green, blue-green, or other. This is a floating mat floating on the surface. So these are, I would say these are pretty thick tufts. This is also in a lake, so it's very slow-moving water, possibly standing. So this is rather unusual to see in a stream, unless you see it in a pool of very slow-moving water in your site.
Just for reference, in case anybody doesn't do this, ½ meter is approximately the length of $3 bills.
Yeah,
So you
that's a
always
good,
have a ruler
yeah.
with you.
yeah, that's a good way of thinking about it. Thank you.
That should be in our field sheets. On our habitat assessments, when we're trying to measure the size of rocks, it's like smaller than a basketball or bigger than a basketball. So I'm all about practical measurement.
Yeah.
Thank you.
So here's an example of submerged mats and tufts of green algae. Although this looks a little bit brown, this is an example of green algae that is covered in a thin layer of brown algae. And you can see that because these are thicker filaments of green algae. If you are able to go into the stream or reach down and you pick up a tuft like this and it doesn't fall apart in your hands, that's an example of green algae. If you see very thin flowing filaments of algae and you're not seeing this thick growth, this is indicative of green algae, whereas floating strands that are very fine are brown filaments. This is an example of submerged tufts and mats of green algae. It's very bright grass green, although there are also plants in here. And we're going to go through how to tell the difference between algae and moss, algae and different aquatic plants as well. So you have some thick, like, tufts of algae here, and you see some long filaments as well. And again, I'm sorry, these are not the best photos, but if you have any questions at the end or want a closer look, this presentation will be available online.
Wow. Sorry, that's supposed to be a much better photo showing chains of brown algae.
There may be another photo later on in this presentation.
That looks really crazy.
This will be fixed by the time we get it online.
Yeah. If you have the Watershed 101 presentation, that same photo is in there. It looks like brown hair, but yeah, well, I don't know what happened there. It didn't look like that at the office.
Could be a Mac versus PC issue.
Maybe.
But we'll probably see an example of this when we go down to the stream. If we do, I'll be sure to point it out and answer any questions that you have about what that would look like. So some examples of brown films. This isn't a whole lot of films, and what we mean by films is just a thin coating. Maybe it doesn't completely cover the rock, but it's just in little sections. So these are all pulled out of the stream, but when they're in the stream, they look a bit lighter in color. So there's just a little bit of algae growing on this rock, and then just a very fine film. If you're able to reach into the stream and pull out a rock, you'll know that these are brown films because they'll be really slimy. If you've ever walked in a stream or you've felt rocks down in there, the really, really slimy stuff are brown, or they're diatoms. So examples of blue-green algae are very obvious. They are either bright turquoise or a little bit darker in color, but they are exactly blue-green. They're not grass green. So you will either see them as floating mats here, and again, this is a picture of either a very wide, slow-moving stream along the bank, or this is a lake right at the edge of the lake. And this is a big, thick, floating mat, and the water itself is really, thick and blue-green in color. So this would be an example of probably plankton of blue-green algae, and then some thick tufts. Down here, it's kind of hard to see, but it looks a little bit different from this photo in that there aren't big, thick tufts. So it looks like there's blue-green algae growing under the surface. And from this photo, I can't really tell you if this is thick or thin, filamentous or not, globular or not, but that's really at your discretion of what you see above or below the water surface. But what you're looking for most is just this blue-green color. And here's an example of surface films, and this applies to any color algae. Again, this is in a lake, and we don't expect to see this in rivers or streams, but if you do, this is something that we would want to know about. It does look kind of grass green, just because of how sunny this photo is, but down here looks a little bit more turquoise, but it's a surface film in that it kind of looks like it's moving with the flow of the water, but it's forming a thin layer on top of the water. And this is an example of something we definitely don't want to see, even in a lake. This is a type of blue-green algae that has the potential to produce toxins. And when you see conditions like this, it doesn't necessarily mean that your water is toxic. This, in some cases, happens naturally if you have a lot of fertilizers or, in this case, maybe some pollution from these boats here. But you do want to be careful. If it's not for your stream monitoring, if you're in a lake like this, try not to stick your hands in there for long periods of time. Maybe don't let your pets go swimming in this either. So here are some pictures of globular blue-green algae that we saw up in the New Jersey Highlands a few years ago. We've only seen this growth form in one site before, but if you see this growth form in any color, this is what globular algae look like. It's very squishy, and when you pick it up, it's squishy, globular. It looks a little bit different than tufts. It's not soft and pillowy. It looks like big old blobs, and it looks kind of gross. But this is actually a really cool type of blue-green algae, at least to me. Again, we've only seen this in the New Jersey Highlands. We've never seen this in any streams in Philly. Yes?
About a week ago, along the Wissahickon, there was one section where there were clumps on a rock here, a rock there, not over the whole bed. It was a brilliant emerald green, just startling.
Oh, wow.
I've never seen it before, and it was only in that one section, maybe half the length of this room.
Oh, wow.
It was a sunny day, but it was just startling. Unfortunately, I couldn't have a camera.
If you see that again,
I was thinking going back there and trying to get a picture of it. It's just, we've blocked the screen for years at a different section, but never saw anything like that. And it wasn't all over everything. It was more individual rock. A rock here, and then a rock a foot away, another rock over here, another rock over here.
Yeah. And in some cases, they kind of stick on one part of the rock and they kind of grow up in these globs. But that's really interesting, especially if you haven't seen that normally in your stream. Also here, there happens to be a lot of, oh, sorry, yes?
I was just curious, you might have mentioned it, and I apologize if I missed it, about the sense of scale of the size of these blobs.
So these are probably, okay, so if we're talking in terms of the size categories that we use when we're measuring rocks, so these would be, these would be about ping pong ball size. These would be about baseball size. These globs would be in between baseball size but smaller than a basketball. So that's that's the way that we
Softball size?
maybe softball size, yeah. But individual globs like this, especially if they're in this stream, there are a lot of thicker mats of diatoms, and you also see some floating strands of diatoms here and here as well. So most of the time you'll see smaller globs like this. If you have a really clear stream, they may be a little bit more obvious. But if you find this to be unusual in your other observations in that middle section, maybe write down how big those globs are, just so that you have some frame of reference.
And a cool thing to know about this type of algae is that it kind of forms like a balloon. So it's hollow inside. So unlike others where the water's going through it, when you pick it up, it literally feels like one of those like squeegee toys you get for like little kids. Yeah, so like if you see over here, it's kind of hard to see, but this is the stem that's kind of attached to the rock, and with the shadow you can tell it's floating because of the hollow space inside of it. So we were all there when we sampled this site, and I remember walking to this because this is something we'd never seen before. And I was like, I hate to give this like description, but I was like, yeah, are those rocks? Do they have skin tags? Like it just looks, it's just protruding that way, and all these different spots of the rock. It didn't seem like there was like a set position of a rock where it would usually be. It was just everywhere. And we're like, whoa, this is like really weird. I don't know if I should be touching this, but this is really squidgy and really fun to play with. Yeah.
So when we go out and monitor algae, we collect samples of different types of macroscopic algae, and our phycologists back at the lab actually take a look and try and identify each of these larger growths of macroscopic algae. I have a colleague who specifically studies blue-green algae, and when I sent him pictures of this, he wanted samples because he thought this might be an undescribed species. The funny thing about algae is that even though they're super widespread, the smaller the organism, the less we actually know about species and taxonomy and their growth forms and their life history. So there's a lot of species that are yet to be described. It's about maybe 10% of total diversity of algae that is actually known and has been collected and formally described. So that's kind of why we're assessing this at a higher level, going by color.
What magnification do you need it to actually see?
It depends on the type of algae. I specifically study diatoms, or this golden brown algae, and they normally range in size from 2 micrometers to 2 millimeters. So I need a really high-powered microscope, anywhere between 100 to 1600 times total magnification. This algae you can see anywhere between 10 to 100 times total magnification, especially if you just take a small section and you spread it out on a microscope slide.
Is algae a species and diatoms are, below that? And you mentioned plankton, and I'm just not sure what algae are.
Most people who study algae don't know what algae are. So algae is a term that doesn't actually reflect any one group. Algae in general just means photosynthetic organisms that get their energy from the sun. They have chloroplasts that allow them to harvest that energy, but they're anything that aren't land plants or aquatic plants. They're single-celled organisms that form colonies or exist on their own. But algae, these are a type of bacteria, and just about every other algae is a eukaryotic algae, so it has a nucleus. But they span the whole field of evolutionary biology, and they're very distantly related to one another. So
Are they all single-celled? Did I hear you say that?
they're all single-celled. They can, if they're forming filaments, multiple cells will form next to each other along a filament. Same with plankton. A lot of planktonic algae form chains because that allows them to float better on the surface. A lot of surface area there. Same with films of blue-green algae or tufts. Forming those colonies help them stick together. But they are single-celled organisms that often form colonies.
Thank you.
So this is a series of photos that we used in our training for algae sampling for the DRWI, just to give an idea of thin mats or films, medium mats, thick mats. Again, this is subjective based on what you're used to seeing, but this was our way of calibrating all of the sites in general because we sample so many different types of sites. So thin mats or films are, they're just like slimy little layers. They may not cover an entire rock. They may just form in little patches. And with green algae, it's actually really hard to see, and you kind of need to get a close look and see if you see any small patches of green films. So a medium type of mat of algae would be covering maybe most of a rock of like brown algae. This is light brown. We lump these all together as brown algae here. And then green, a medium mat at least the way that we were calibrating it, was about 0.5 millimeter to 3 millimeters, so just starting to form a mat. It's a little thicker than a film, but it's not a really heavy coating. Thick mats of algae are either in these like globular forms or covering most of a rock, but when it's in the stream, it looks very thick. Like this is a type of green algae covered in brown algae, and again, the green algae are thicker and they hold up in your hands. This brown algae, you can hardly even see that there's a rock because there's so much brown algae growing on it. So some short filaments. If you're comparing filaments versus mats, a mat would be covering most of or an entire surface of a rock or other types of substrate, but filaments are flowing. So here's some filaments of green algae here, and when they're in the water, you'll see them as sort of like thick strands of hair. Brown algae, and it says reddish here. If you see red algae, that would go in your other category. They look like very fine hairs. Longer filaments down here, again, we'll probably see these in the stream. They're very fine hair. You pick it up, breaks apart in your hands. And these are long filaments that are different than mats in that you can see them flowing versus just thick tufts growing on substrate. With green algae, long filaments in our case would be anything longer than the length of $3 bills next to each other. And when they're underwater, they may look vaguely green because they might have brown algae covering them as epiphytes, so they're growing on top of the green algae. They may look like these really long strands if this is a very thin stream, it's almost like a ditch. Or if you pick a rock out of the stream, you can see these longer filaments here. So here's what we're talking about when we talk about rock snot or Didymosphenia. So you may have seen this picture that when we were putting this presentation together, I asked Catherine and Amanda to find this guy because in almost every presentation that mentions Didymosphenia or rock snot, this guy shows up in the picture. If you do a Google search for rock snot, he comes up as one of the top hits. It's very unfortunate that he's in this stream because while this isn't toxic, it is considered to be a nuisance species because it's so thick. And it looks like it's gross. It looks like dirty, wet wool, dirty, wet toilet paper, and it really holds up in your hands. It feels very thick. It's also kind of slimy. Again, this is very rare that you would see this in any of your streams, and certainly not in as much as this. This type of diatom, or rock snot, when it grows in colonies, is considered invasive. It originated in Australia and has since moved almost worldwide. It's shown up in cold, fast-flowing water throughout the United States, and it's unclear when it was first introduced to the United States, but it's believed that a big reason why this is an invasive species is because of, fishermen. If you have waders, especially if they're felt bottom waders, diatoms, again, are really small, even though these colonies are really thick. If you're not washing and scrubbing away all of the algae and the stuff that's on your waders, and you go from stream to stream, you may be transporting this rock snot. Even if you don't have felt bottom waders, we, as best practice, if we're going from stream to stream, as long as they're not part of the same original river, we assume that there are going to be different colonies of algae. And we don't want to introduce any species, whether or not they're invasive. So just as our protocol, we scrub our waders, even the rubber-bottomed ones, anything that has gotten into the water. Any questions about rock snot?
Are they considered a nuisance in Australia?
Yes, and it's actually a big problem, especially for fish and any other organism that lives in the water that would normally eat algae.
You can see from this photo on the left, he's standing in a sea of it, like you can't even see that there's a stream there. It's just dynamo. And so you imagine there's hardly got to be anything else living in that creek after a while.
Definitely. It's completely blocked out the sunlight because of how thick it is, so it's really difficult for other organisms to live below the surface.
Well, just out of curiosity, how did it evolve in such a way that there's no checks on it, even where it originated in Australia?
Like a lot of other invasive species, it's adapted to being able to grow really, really fast, especially in these big colonies, and they can spread a lot. And because the colonies have so much mucus around them, it's really hard for fish or other organisms to eat this because they don't want to be eating a ton of mucus. So that's kind of a defense mechanism, and it allows them to spread a lot.
So the environment here, temperature, water here in this area, is it suitable for this, or it's not here yet, or it's just simply not suitable, that's why we're not seeing it?
It's been found in very, very small quantities in this watershed, up farther north than where we sample. It has not.
And in the main stem.
And in the main stem.
Yeah.
It's not made its way to smaller streams, but just as a precaution, we want to be careful that we are not.
We don't want to
going
be responsible for contaminating somebody else's stream.
Yeah.
Yeah.
But the conditions here are not really conducive to dynamo growing.
Is there some measures taken to, for example, at least stop growing that?
It's mostly photos.
I understand that
Just like a couple more
nature can kind of fight against these kind of invasive plants, but are there measures that human can do?
That's a really good question. As far as I know, there aren't any really good successful strategies other than preventing the spread. I don't know what sort of chemicals or anything are being used to.
We just clean our waders with dish soap, and we use like
Dish soap and
environmentally friendly dish soap. It's just a matter of cleaning them.
yeah.
Does completely drying waders of roots work?
Yeah, but that's why, yeah, so we clean them and we dry them, but that's part of the reason why felt bottom waders are starting to be banned, because you never can really quite get them quite dry, and things just stay up in that felt. So it's unfortunate because they're a lot easier to walk around these slimy streams on, but they're really hard to clean.
So one thing that they mentioned that, so you guys really shouldn't have to worry about this too much because you guys are going to the same site every time, you're in the
Yeah.
same watershed every time. This is really just if you're stepping out of your watershed. But we're gonna, everyone hold their questions here for now. They're gonna run through a couple more slides. We wanna make sure we have time to get down to the stream, and you guys will see examples of some of these things down there, so.
Not this.
Not this, yeah, hopefully not.
So I think we just have a few more slides left to go.
Some that show plants.
Okay, so sometimes it's difficult to tell the difference between algae and other plants like moss or aquatic plants. So we'll go through and try and look for plants or moss in the stream. But what you're really looking for when you're comparing plants to algae are leaves with this is algae growing on them.
No,
Really,
that was the one that had. So, case in point, I freaked out because I saw that and I touched it and it felt like wet wool, and I thought, oh my god, dynamo. We brought it back to the lab and it was actually insect egg casings. So, you never know. There's a lot of things that look like algae that are not.
yeah. And same with these plants here. This may be a little bit difficult to tell whether it's filamentous algae or not. But this actually has flowers growing out of it, so this is an example of a plant. You may not always see flowers, but it's good to just try and pick things up in your hands and look for leaves or different parts that would indicate a plant. So another thing that is really hard to tell the difference between is filamentous algae, and moss. So a really good way to tell the difference is to pull it out of the stream. You'll be able to feel it in your hands. You'll be able to pick off individual pieces of moss, and you wouldn't be able to do that with algae. All right. And then these are examples of aquatic plants here that have a brown layer of brown algae growing on them, but you can tell that these are aquatic plants, especially if you pull them out of the stream. Likewise with these, they're sticking out of the water. These are aquatic plants, and these are thick mats of green algae. We are actually going to skip these comparison sites for now, but just as an example, this is a very low amount of algae, and this is a really thick mat, comparing the two. And here's just an example of a site that we go through. This is an area that has been cleared of algae. It's been scrubbed off of a rock, and this is what is considered normal conditions for this stream. So you can really see the difference in growth, and especially if you're getting really close to the stream, you can see that there is some thickness of algae. And just as acknowledgments, I just want to thank everybody from the DRWI team and our support. If you don't already, follow us on Twitter, check out our websites, and again, this presentation will be posted online. So this went on a little bit longer than I expected, so I apologize. If you have any questions for me in general, feel free to ask me once we get down to the stream. But we want to make sure that you get some field time before the end of the workshop.
So you guys can move your stuff in here if you want. We'll be able to get back up. We don't have a ton of time, so we're going to head straight back.
So how many of you guys see long flowing filaments of brown algae? What's long again? Well, long or short? Short. Short? Probably less than half a meter? Definitely. If anybody wants to reach in and try and pick some up in your hands,
Can you repeat what you said? Because some of us were just arriving.
yeah. We're looking for examples of long flowing brown algae filaments. So if anybody, if you guys want to pick it up in your hands, kind of squish it around between your fingers.
Because it breaks apart, it's brown?
Breaks apart.
Yes. Yeah. So these are filaments of diatoms.
Okay.
And they flow these. Yeah. Those would be diatoms. And if it doesn't break apart, that's a type of green algae.
Okay.
Yeah. So when you're doing your assessment, you want to record length and whether you're seeing filaments
this is
or
just breaking
mats,
apart. So
or maybe both.
So it may be green and brown because it's kind of.
And so you can see these filaments are flowing through the stream. They're kind of moving with the flow of the water.
So, do you consider those as filaments, or the tufts submerge the tufts?
I would consider these filaments. They're really short.
Yeah.
And
So
if you're unsure, there's no wrong answer really. We're less interested about in the growth form as we are the amount of growth and the color. Yeah. These are just.
So when you actually touch them, they broke apart. They're still alive, right? They just happen to just go downstream and somehow attach to something.
Yeah, you're just breaking up the colony really, and they can reform the colonies. So right here would be an example of both submerged mat and filamentous brown algae. So both growth forms.
This would be medium coating?
I should be downstream of you. I'm sorry. That's really bad.
No, it's okay. So I just scrubbed away this little part right here, and I can actually move a little bit more of this around. So if you look in the water, that scrubbed away area, I would say that's like probably a medium growth. If I didn't have any kind of baseline, or if my baseline meant a low amount of algae, I would consider that pretty medium.
Sorry about that.
Okay.
Let's see.
It looks like algae.
Sorry.
Oh, look at that.
A lot of sediment settled in the algae. Yes, not actually raw algae.
Yeah, and you can usually tell the difference. It'll look really grainy, and it'll.
And you can like dust it off.
Yeah. So this is green algae.
Green
here.
algae, yeah.
Yeah. If you spread it around in your fingers, you can't see any kinds of leaves or. It doesn't feel like moss.
Right.
green
so
algae?
These are
She's
algae
taking a
filaments. If we
Green
stick this
algae.
back in
Green
the water,
algae.
we can see
You could
the
see before,
filaments
when you were
flowing.
picking it up, you could see that it was.
So that's green algae, you think?
This is green algae. Yeah, it's grass green in color.
But it falls, kind of falls apart and it feels kind of munchy.
Here's some more green algae. I don't know if anybody wants to touch it or pass
Kind
it
of
around, but
filaments.
if you pick it up in your hands, it does break apart a little bit, but it's grass green instead of brown. Oh, yeah.
So you would say this would be a baseline?
There you go.
If I had never
You can
been
take
here
it, or you
before,
can photograph it, whatever you want to do.
Yeah, I'm videoing. Oh, yeah.
there's no algae,
Probably good
I
for
would
you, too.
consider this medium,
You good?
Yeah.
medium amount
Okay.
of growth. So if I was thinking my baseline is very low amounts of algae, this would be on the towards the higher end for me.
Something on that I want to say to some of the speakers. I think it'd be good for everybody. This first month, don't worry about filling out the assessment. Use this first month as a way just to take a look at it. Look back at some of the photos you've taken of algae in the past. Don't jump to conclusions on this first one, and maybe just get a feel for it this first time. Next month or in June, you'll have a good idea what the baseline is.
Should we start taking a fourth picture regularly, just looking down?
Yeah, so what those photos she was talking about, this is all in addition to that the observational monitoring you're doing, the 3 photos you're taking. So this is all going to be in addition to that. The 4 photos is talking about specifically taking photos of algae.
No, I mean, should we add one to our regular 3,
Yeah, you should
and make it down?
Right, you can add 4.
So make it across, up, down, stream, and down.
And then photos of algae.
Yeah.
I
So if you're
misspoke
taking a photo
earlier.
directly down, it's because you're looking at
look to you so we can
Yeah.
pull
You
you
just
back
take a
upstream if you
picture of the stream bed, unless you're specifically looking at some algae.
Oh, okay.
And if you're able to try to avoid glares from the sun because it makes it a little bit harder to see the algae.
Mine are not actually bleeding.
Yes.
No, I'm just trying to hear what she said.
Probably should have.
Basically, yeah, that's what I'm saying is that you wait, give it a month, or maybe the next month, then you'll have a better idea of what that 5 and your
But I think
baseline's
they'll
going to
be
be.
fine.
So it's all dependent on individual strain.
Exactly.
Yeah.
Exactly.
So this is some filamentous green algae. It's not growing on a rock, but it's actually growing off of this leaf. There's a little
Okay.
worm in there. But it forms really fine filaments that you can actually break apart in your hands. But because of the color, you know it's green algae. Even though it's slimy.
Do all those leaves have it on it, and you can't really see it till you pull it out, or did you just find one that?
I just happened to see this glob of green algae
Oh,
here.
look at that.
Yeah. So this is a short filament of green algae. I just picked it out of the stream growing on the leaf.
Oh, it was on the leaf, okay.
Pick it up, it look like much browner, and then after you smear.
It was actually really bright. Yeah. Yeah, it's growing off of this leaf here.
That's kind of like what I saw on these rocks, except it was all a really bright green, almost like a shirt, but even
okay.
greener. Yeah. So that wouldn't be blue-green, that's just green, right?
Yeah, grass green is green algae. If it has any kind of blue tinge to it, or it looks turquoise, that would be blue-green algae. But if you spread this around, this is grass green. This is very bright. It's really hard to tell. Usually when green algae are really slimy like this, this This is a type of algae. It's called conjugating green algae. If you've ever heard of Spirogyra, that's a type of slimy green algae.
Nourishing,
Yeah.
This is basically normal base flows for this time.
So you said the brown sometimes grows on this, or this sometimes grows on the brown?
With really slimy green algae, diatoms don't really like growing on that. They kind of slip right off. But any sort of thicker, coarser green algae, diatoms love to attach themselves to that. And then here on my wrist are, this is all diatoms here. This little brown films. Yeah.
Some smaller, like minnow type fish.
I think that's really good.
I know, every time we come to one of these things, like,
But this does look like our brown.
Yeah.
So if you dig up, if you dig up some individual rocks, you'll actually see they have the, a lot of them have the brown diatoms on it too. Kind of, it's like how she was talking about. That's kind of like the base layer of a lot of.
I think
the some of the filamentous algae building on.
So you have the diatoms first and then the...
Yeah, a lot of times that's how it
the
is.
algae, the filament algae grows on top of it.
Yeah, it kind of creates that like biofilm that she was talking about earlier.
Yeah. So these would be short fibers. Yeah, so
When I wore waders, somebody lent me some. So it was out in the middle of a fairly deep part of the ocean. And you can really start to lose your balance on that because they're hard to move anyway. If a rock shifts under you, and it's up to here, you can't move your leg quickly. Anyway. I got very nervous. I wasn't sure I could get back to the hole.
yeah, that's actually kind of my question, because you mentioned that means after the prime season, they just die out, but then they have like for plants, they go dormant, right?
Yeah.
Are they also go dormant and then just still stick on the rock or the bottom of the crate?
Yes. And because they're so small, it's really hard. It's sometimes really hard to find these dormant cells, but they will form resting cells or ways for them to kind of hibernate.
So when you say they're single cell, just saying they're globular,
Yeah. So in the winter, that might be the ecology of that specific algae.
Okay.
So that might be like how they survive in the winter. They stick on and they can just be like dormant. They can be dormant in the winter and they're still alive. But if they were dead, they would not be. Yes. It would be cleared out just because there's nothing holding. There's nothing holding it. So if it's not alive, it's not like aware of that. Yes. Yeah. Yeah, this is really helpful. You're really
Now you can
appreciative.
see the difference. It used to be brown, brown.
Yeah, like I really like about this visual assessment is not to interpret it, it's to know any differences and how like any changes. Because like when you start with interpretation first without understanding what it is, that's like a real easy path to misinterpret what's going on. And like that obviously is not going to go in like a really long sustainable path of like how to change it, how to fix it, or anything like that.
So picture probably is the best way to see.
So if you notice something like globular, the blue-green thing, that was our first time ever seeing it. And if you see how we took pictures of it inside of the water and outside of the water, that's a great way to take baseline photos. Since you guys are the first time doing this and like taking notes of the algae, it's a great way to take photos. Taking photos is a great way to know like what you see and how much of it. And if you say like, oh, like, in this photo I gave it a 4, you can look back in years after, like, oh, so this is what I thought it was a 4, so now this is my standard, and now I know what like a 9 is in my idea, and like what a 5 is, and a 6. So it's like a way to like calibrate yourself. Yeah.
I can get this to a microscope. I want to take some pictures, maybe upload to you guys, so you can look at it.
Oh yeah, I'm sorry. Yeah.
No, finish.
Oh, okay. So when I take my photos, I have a life case. I have an iPhone, and I have a life case that is waterproof. And like those pictures that of the globular algae, I took those photos with using my iPhone because the case is waterproof. I just dumped it in and I was like, I'm gonna believe you life-proof and I hope to God you're great. And then you tested this.
Yeah, it's nice something to watch out for.
And like I was talking to my colleagues, like,
But yeah, this is helpful for me because it's like algae is just a word, but it's like some are bacteria, some are whatever.
Also red algae, that are freshwater algae. There are so many other groups that
Yeah, they don't have that vascular structure that a macrophyte would. So it's like, yeah, but a lot of people kind of lump them all into that one
I've had
category.
that issue, like, because I didn't.
But they're really not classed as plants, right? They're not.
beds of green algae
they're
that are floating.
part of
They kind of look similar to some of the actual aquatic plants that.
like a weird distant cousin of plants.
Someone, was like, I have this really thick bed of vegetation, and I was like, I think it's algae, and he was like, no, I think it's not, and I'm like, I don't know.
Depending on the species, some are able to
you're good.
so they can live in environments where there's not a lot of nitrogen. Others are able to withstand drying out, like desiccation. So individual species of algae can live on like wet cave walls, or they can live in areas like blue-green algae, for example, can live in the world's driest desert. As long as there's just a tiny bit of water, they can stay dormant for long periods of time. That's not the case with all algae, but individual species can have really specialized habitats like that.
I think so. One I got it or something. I think they might have it my site. Like I was a little expensive,
Definitely, yeah.
and Matt would be new. Does that mean more? It's expensive. Talking about the area that it's covering, or the fact that it's thicker than that. But you can see that on that green filament, there are some brown flecks. The diatoms will grow on there. Maybe. So sometimes you'll have both. Oops, sorry.
So the rock has, at first it gets the diatoms and then the algae grows on top of attaches to the diatoms?
No, not necessarily. There could be a rock that has green filaments, but no diatoms. Pretty much diatoms will grow on anything.
What does we can't see them, what does it look like when there is a, diatom without the algae?
The diatoms are going to be everything that's brown and slimy on this rock.
All right, so
This is all
that's the
diatoms.
slime that doesn't have the diatoms on it.
Yeah, and this one has a little bit more coverage, so you can kind of see all of this brown, like, goo.
If it's brown and it doesn't have filaments, then it's probably
Yeah, but sometimes the diatoms will form chains, and so that's why we do have a category on there for brown filaments, because you can see these little short bits here that are floating. They're brown, and
But they're diatoms,
but
they're not the algae.
Right, so
So
that's
would
where
be
it helps to kind of touch it if you can, because the diatom chains, and sometimes they can get quite long. Unfortunately, I do not know what happened to that photo that looked all pixelized, but you can really see in
And the
It
phosphate
looked, in the water, they looked like they moved in a similar way, but once you pick them up, they just fall apart.
All right, so is that on the thing I noticed, over 5/10 of a centimeter and over and under?
Yeah, and
And that would be pretty much a diatom if it's under 5/10?
not necessarily. That was a over or under half a meter, because some
meter?
green
All right.
some
I
green
thought it was
filaments can get quite long, and really that's sort of just an arbitrary thing that we breaking point that we put in to sort of differentiate long versus short.
All right.
I mean, if we asked people to get in and measure it,
I thought it was a
sorry,
centimeter. to be
it's a half
truthful, so
meter.
just looking at it.
Yeah, and the length is going to vary a lot based on disturbance. Like I probably stepped on this one and rubbed half of it off. And the more I'm playing with it, the more I'm breaking up the here's a nice long one.
The algae.
Yeah. I don't know if it's hard to see. This one right where my finger is.
Yeah.
So now the question is that diatoms, or is that a green filamentous strand that has diatoms growing on it? So I'm just going to push it with my finger and it just fell right apart.
So this one up here,
So that means it's
that's a filamented algae, or is that a diatom?
This one is
Oh, it's a
filamentous.
green?
Yeah. But it has
The
some...
diatoms giving it the brown appearance?
Exactly.
All right.
And we,
it's hard, we can't always tell just from looking at it too. Like, I've been sampling algae for a few years now, and I still need to be like, is it? And it's hard to tell just by looking.
So if you use a microscope, you can see better? Or not?
Yeah, I mean, but that's not it. When I'm doing algae sampling, I scrub the rocks, I get the juice, and I send it to the lab. Like, I'm not a microscope person, and we're not asking you guys to be that either. So that's why we tried to be very descriptive and like circle as many of these as make sense, and we can figure it out.
I guess the gist, or what to get from all this is if there's a huge amount of change, it's not good. Right. One way or the other.
Well, yeah, and the change that you see doesn't necessarily mean that something bad is happening.
Right.
It could be seasonal change that's just normal.
Right.
So that's why, you guys already have a sampling schedule of monthly, and if you didn't, we do our algae sampling May to June, ideally. Last year, for example, was very rainy, so every time we got a large rainstorm, which happened to be seemingly every weekend, which was irritating, not only because it ruined your weekend, but also our protocol is we need to wait 7 to 10 days before we can go out because everything has been scoured.
Right.
And so we would get a rainstorm on the weekend and that would wash out our whole sampling week. So sometimes our season ends up going June, July, August.
We have a narrow window, so.
Yeah, but then that's good then because it's on your sheet. You know how much did it rain yesterday? So is this unusual or is it just because it got washed away? And those kinds of things differ. You guys don't need to worry about that because you work here, but we spend a lot of time looking at the USGS gauge stations so that we can see how much rain an area has gotten. Not just the precipitation, but we look at the stream flow, the base flow. In the Poconos and the upper Lehigh areas, 1 inch of rain, probably not going to do so much, but you guys know down here, 1 inch of rain spikes everything. So it's going to be a different threshold for whether or not we go out, depending on where we need to go.
So just to try to get through this, if you put it on your fingers and it falls apart, it's probably not.
Yeah, it's probably a strain of diatoms. So this here, if you rub it hard enough, it's going to fall apart, but it doesn't just break up in your hands. This is a filament of green. See, I did break these. But these, and I wish I could find a longer strand of brown, but this sort of strands that are flowing around at the edge, can you guys see that? Yeah. If I try to grab it, it just dissolves. Yeah. Yeah, you'll be able to feel in your hand, and you guys can touch that. You can feel in your hand that there's something there.
Do they ever attach to living toes?
Probably, yeah. Slow moving turtles I've seen with like little things on bits on them. Yeah.
I mean, I know they're not attached to the mud.
Yeah.
Get in and out of the water, but
Anything that stays still on it, I guess. Yeah. Is it really
Who said that? Who said
like if you're in a forest,